two?
Just one.
Okay. So, welcome to my talk. I'm going to quickly present my project, which I'm doing at the Ludwig-Maximilians-Universität in Munich, and our cooperation is with Era 7, but we are still figuring out how this is going to work out. But so this project is entirely biological from the beginning. So what I'm working on is selection on quantitative traits. The quantitative traits are traits which are determined by a large number of genes, by contributions, bigger or smaller, by a lot of loci. And one such trait is the so-called Chill-Coma Recovery Time. I will talk about what it is. It is a phenotype that people use in Drosophila genetics. So temperature is a very important environmental variable, and Drosophila melanogaster is a species that has done really well in adapting to diverse environmental conditions, and this includes very diverse thermal niches. It has a network of populations that are present on every continent, and there is huge genetic variation that underlies these thermal adaptations. So this has been studied extensively from both ecological and evolutionary perspectives. Chill-Coma recovery time is a phenotype that people in quantitative genetics use because it is very easy to make experiments and it's very easy to measure. So insects in general fall into something called a chill-coma, which is defined as the ceasement of movement after a certain threshold of temperature. They just stop moving, and in this state, provided there's no like ice cold damage, is completely reversible. They survive this way. Not a lot is known about the actual physiological reasons why this happens, but it's clear that this is a very complex trait. The way we measure this Chill-Coma recovery is by looking at how long it takes for Drosophila flies to wake up. So you just put them in the fridge, and then you take them out, and then you wait for them to wake up to start standing on their legs. And I'm not doing any experiments of this kind, but some of the people in our lab do, and basically my project is more about data analysis, so I will talk about that. So one thing that's important is that this resistance to cold varies markedly between different populations of Drosophila. And the other thing that's important is people have done a lot of selection experiments with this phenotype, and they have looked at how if you try to get the flies used to the cold, how it affects different other life history traits such as development time or viability, and they showed that it was possible to just select specifically for this trait. The other thing that people have done a lot of research on, especially in Trudy McKay's lab in Edinburgh, was to uncover what the genotype-phenotype map is for these different complex traits. And they have done genome-wide association studies to identify which variations are associated, which single nucleotide polymorphisms are associated with these traits. And since now we have, for Drosophila melanogaster, we have about 200 fully sequenced genomes already, and many more are on the way. And they are completely sequenced and to a high coverage. Now we can actually use this knowledge on which SNPs are associated to some quantitative trait, in my case Chill-Coma Recovery Time, and look at how they differ among populations from different environments. So this paper here was published in last year, and its focus was on the genetic architecture of 3 complex traits in Drosophila: starvation resistance, startle response, and Chill-Coma Recovery Time. And they actually did something called extreme QTL mapping. So these are the images from their paper, where they took 40 inbred lines that were sequenced in a previous project, and they did a round-robin mixing and mated them randomly for 70 generations to create another population which was now outbred. And then they took these flies, phenotyped them for these 3 traits, and then took the 15% from the lower and from the upper tail of the distribution, and then they resequenced those 2 pools. They pulled those together and resequenced them. And then what they wanted to know was if this sequence, for these SNPs, the allele frequency was significantly different between the low pool, the blue pool, and the red pool. And if it was significant on a genome-wide level, over a certain threshold, then they said, okay, this polymorphism is significantly associated to this particular trait. So what I did is I took this information from their paper, and I took sequences that we have available from all these different populations, and then I chose the 2 populations where the climates were the lower temperatures. So these are average minimums and average maximums in all these different populations. And I chose the population here from Rwanda, and here I have 2 populations from France and from the Netherlands. And my hypothesis is that at these sites, at these SNPs that are associated with Chill-Coma recovery, I will be able to find significant differences between the African and the European populations. So what I did is, this is a software developed at our lab where you can easily retrieve the sequences. I used a pretty stringent quality score, and then I concatenated all the SNPs together into one pseudo alignment to decide how many ends to allow. So because this is next generation sequencing data, you can see that some lines, so every line is one individual. You can see that there's a lot of missing data. This is important for later analysis. And what I used was Wright's F statistics, and these statistics tell us basically how much variation we have within a particular population compared to between different populations. And what this tells you is if you have the greater percentage of variation is found within populations, then that means they are diverging, and if they are diverging at specific sites, then that suggests to you, that can be a good suggestion that there is selection on this phenotype. So I used this, there's a lot of ways to calculate these F statistics. I used the method by Weir and Cochran, which is good because it can count for different sample sizes, such as if you have one population that's smaller and one that is bigger, that it's not going to influence your F statistics calculations. And what I found was, if you look here, so this is the distribution of FST values for, so these gray and blue ones are different ways to represent the genomic background. These are like all genomic SNPs, so this was done just for the comparison, and this is my sample. So you can see that overall that there is a significant difference between the two distributions, which tells us that there is something going on. What you can do also is there is a lot of information other than just the sequences, so you can look at these SNPs more closely and see exactly where they map, on which genes, and which regions of the genes are they in coding or non-coding regions, and so on. And so this is a more recent. This is just a result that I got when I performed a lot of quality controls at the end, and there's still a significant difference between these. I don't show the p-values because they are really very small. They're like on the order of 10 to the minus 16. So it seems even if you subcategorize the sample into like genic SNPs or just the SNPs that are in the third codon position, there's still a significant difference. So what I want to do now later is see one thing in particular, and that is whether we can find preferentially the SNPs that are associated with lower cold tolerance or the higher cold tolerance. If we can find those preferentially in Europe, those that are supposedly better adapted, because this is something that people have already done in humans with height. And also I want to make sure that the result is okay, that it's not confounded by ancestry or other ascertainment biases. Yes, also what I want to do is estimate the strength of selection. If there indeed is selection, I want to know what the selection coefficient is. Look maybe at the biology of the genes that these SNPs map to, and at the end look at genetic networks and see whether there is any patterns in these genes. So thank you